Now that we've looked at different organelles and the adaptations that they have to be successful in supporting the cells that they are a part of, we want to turn our attention to looking at specialized cells and cells that have specific functions to carry out different functions. In the creation of a new offspring through sexual reproduction, there is the fusing of sperm and egg, and this process is known as fertilization, and it's the fusion of the male sperm and the female gamete, and that produces one single fertilized cell called a zygote. And so in humans, sperm have 23 chromosomes, females in the egg have also 23 chromosomes, and when they combine, it makes a full set, a diploid set of 46 chromosomes in this zygote. This single cell then divides repeatedly through the process of mitosis, in which each of those cells are identical to one another. Mitosis produces identical cell copies. And so we have a growing number of cells that are all identical. And initially, those cells are unspecialized. They are all of the same type. They don't have specific functions. But as that zygote and that fetus continues to develop and becomes a baby after birth and then grows into adolescence and an adult, those cells become specialized and they have different functions and different roles. And there's about 220 distinctive specialized cell types in humans. What controls or causes these cells to become different is all dependent on genes and DNA. Because each one of those cells originated from the zygote and they're all identical, every cell has the same type and amount of DNA. Obviously, it's possible for some mutations to take place, but for the most part, every cell is identical. It has the same copies of DNA. And so then what allows one cell to be different from another? What allows a cell to become cardiac tissue versus muscle tissue? It's all dependent on the genes and the expression of those genes, essentially switching on particular genes in one cell or cell type, but not in another. It's kind of like a light switch, on or off. It's very simplified, but that's essentially the process. And so different cell types are caused by the expression of only some genes. Different cells are going to have expression of different genes. Gradients of the chemical signals that are responsible for turning on and off these genes can indicate the position of the cells within the embryo and help to determine the differentiation pathway, which type of cells it becomes for that particular cell. And so if the chemical signal is really strong, it can activate some genes, and at a different physical location within the embryo, that same chemical signal will be really weak, not very present, and so then that may activate different genes. And this is very much an oversimplification of the process. You can take entire courses in college looking at the expression of genes in embryo development, but this is kind of an overall overview of how that takes place. Chemical gradients then are the regulators of gene expression and can impact the expression of those genes. An example of this is retinoic acid, which helps to guide the development of forelimbs, the pancreas, lungs, kidneys, among other organs and structures within the organism. The overall process of cells becoming specialized is referred to as cell differentiation. And differentiation just essentially means difference, so cells becoming different. That is the phrase that describes cells becoming different. Before cells become differentiated and specialized, they are referred to as stem cells. And stem cells are undifferentiated cells. They can divide repeatedly. They have the capability to do that. And once they do begin to differentiate and become specialized, they are no longer stem cells. They begin down a path that's an irreversible path that then allows them to become a particular type of cell. Human adults do actually have stem cells. It's not just a developing fetus that has stem cells. We find them in bone marrow, within skin cells, liver cells, and the purpose of these different types of stem cells is really to provide regeneration and repair, to be able to repair damage. And so each stem cell has a niche. It's a location of the stem cell within the tissue. It's where it resides. And it provides a microenvironment. And within that environment, there are the conditions necessary for the stem cells to be able to remain undifferentiated so that they can become whichever type of cell within reason is necessary, and it allows them to remain undifferentiated for long periods of time, but then to divide and differentiate rapidly when needed. And so some good examples would be found in the striated skeletal muscle cells that we'll look at even a little bit more in detail coming up. They remain inactive unless there's a muscle injury. And so if there's a muscle injury, then new cells would be necessary. And so then there'd be activation to replace the damaged tissue and to regenerate that tissue after damage. Bone marrow and hair follicles, other examples, they provide microenvironments that allows for continual proliferation and differentiation through the production of red blood cells or hair growth. That's a continual process. It's always activated, a little bit different from the striated muscle cells, and that those are only activated when needed. And so the potential of stem cells and the use of stem cells is actually an area of a lot of research due to the possibility of the production of human organs. One of the leading causes of human death is organ transplant, and waiting for organ transplants. Not only is it difficult to find a donor, it's also really difficult to find a match. And even with finding a match, it's sometimes the body is still rejecting the organ. And so the potential to be able to, for one person to have some cells extracted and to be able to grow tissue and organs from those extracted cells by turning them into stem cells has mind-boggling awesomeness and really cool potential that could save lots of lives. Another aspect that you maybe have seen in the news is the lab-grown of tissue, of meat, and so rather than an animal growing, it would be the production of muscle fiber within the lab. And we're starting to see more and more companies do this. An actual hamburger has been produced. Although it was very costly, over time this may become more and more common and more affordable. There are three main types of stem cells, and the first type is called totipotent. And these are found in the early stages of embryos, and they can differentiate into any cell type. They're the completely undifferentiated stem cells, so they have the ability to become any type. The second type is a little bit more specialized, and these are called pluripotent. And these would be found in the embryo, and they can differentiate into a range of cells, but they already have a little bit of specialization, and so they cannot become just any cell type. So they're a little bit more restricted than the totipotent. The last level is the multipotent, and these are stem cells that we find in adults. They can differentiate into several types of mature cells, but they lack the ability of what we see with the pluri or the totipotent. And a good example would be in stem cells that are in the bone marrow, can create different types of blood cells. They have that capability, and so they can become specialized in a little bit different way, but it's a more narrow scope or possibility of their potentials. We'll now turn our attention to examining a couple of specific examples of cells that are specialized. And in doing so, we want to first look at the size of cells, because as we've discussed previously in the course, cells have a typical size. For eukaryotes, it's about 10 to 100 microns. And most cells are of those sizes for some very specific reasons we'll see here in a moment. Sometimes we see cells that don't really fit that model. For example, a sperm cell, although the body of it is very small and narrow, its tail is really long. It's about 50 microliters in length. The egg cell is much larger. Red blood cells are on the smaller size. White blood cells are bigger, particularly when activated. And then cerebellum neuron cells are smaller and have really long axons, really long tails to be able to send nerve signals. And so the size of cells are specifically adapted to its particular function. And there's a reason for them being small or large in comparison to what we see overall on average for cell size. Metabolism is the process in which cells carry out reactions in order to be able to survive and carry out their functions that they have adapted to. For example, the replication of DNA before the cell divides would be an example of a reaction that occurs within cells before the division takes place. And the rate of those reactions, the rate of metabolism, is proportional to the volume of the cell. Cells are three-dimensional shapes, and so they have an external surface area and then also an internal volume. And substances used in reactions must be able to be absorbed and waste products be able to be excreted. And how quickly that can take place, the movement in or out across the plasma membrane, is dependent on the ratio of the surface area to the volume. And the higher the ratio, the more surface area there is compared to the volume, the higher of that ratio, the more efficient and essentially faster molecules and different products can be absorbed or excreted from the cell. And so if the surface area to volume ratio is too small, substance will not be able to move or enter the cell fast enough. Waste would pile up, cells may overheat because reactions produce heat faster than what can be lost. And so because of this, we see cells be typically of this particular size, that for eukaryotes 10 to about 100 microns in size. Yes, we have exceptions where they're smaller or larger than that, but that's the typical size that we see of cells for this exact reason. Some cells have specific adaptations that help to increase the surface area to volume ratio, and one such example would be found in red blood cells. The purpose of the red blood cells is to be able to transport oxygen to tissue throughout the body after being oxygenated in the lungs and then sent back to the heart, at least in humans and other mammals and eukaryotes. The shape and the size of the red blood cells provides a really large surface area to volume ratio. It's about an 8 micrometer disc shape, and it's biconcave, meaning it's like there's a thumbprint on each side of that disc. And you probably have seen pictures, and we have a picture here of a red blood cell. And this shape provides maximum distance from any part of the cytoplasm in order for oxygen and other molecules to be able to diffuse across the plasma membrane. It helps to increase that, the shape helps to increase that surface area to volume ratio to be as great as possible, because moving substances in and out of the red blood cells is essential to its function. The second example is proximal convoluted tubule cells. And these cells are helpful in that they reabsorb substances from the glomerular filtrate. And this filtration is a process that filters the blood plasma through the filtration membrane, and it's the first step in urine formation, where the kidneys are removing toxins from the blood plasma. And so this process takes place near the kidneys or in the kidneys, receives large volumes of fluid to be filtered out of the blood, and some of that filtrate is going to be reabsorbed, glucose for example. And so the wall of it is only one cell thick, and that allows molecules to be able to pass across the membrane very quickly, easily. The channel and the pump proteins that are within that membrane are used so that only substances, specific substances, are reabsorbed and leaves waste products, urea specifically, out. The apical membrane, the membrane on the outside, has a large number of microvilli, and there are small tubular projections that help to increase the surface area, and the basal membrane has infoldings. The alveoli or alveolus are air sacs that provide large surface area for gas exchange that takes place through the process of diffusion. They're one cell thick, and they have a protective layer of cells around them. Primary purpose is to exchange oxygen and carbon dioxide in and out. So oxygen we need in, carbon dioxide we need to be able to release out. And that happens through diffusion between the air in the alveoli and the blood that's flowing in the adjacent capillaries. Concentration gradients must be established in order to allow the diffusion to be able to take place. The alveolus is singular, alveoli is plural. It's lined by a thin film of moisture, and that allows the oxygen to be able to dissolve and diffuse into the blood and allow CO2, carbon dioxide, to be able to be exhaled. In order to ensure that this process happens, the surface tension needs to be reduced as much as possible. Otherwise, the alveoli could collapse. And how this takes place, or how this is facilitated, is phospholipids from the lamellary bodies with proteins, or in combination with proteins, create a single layer on the outer surface of the film of moisture, and the tails from those phospholipids face towards the air, and that helps to reduce the surface tension so that the gases can be exchanged. There are two types of alveoli. The first is type 1 pneumocytes, and they are adapted for diffusion of oxygen and carbon dioxide. It's passive, so there's no mitochondria that's needed for them. They are very small in cell volume. Their thickness is 0.15 micrometers, so really tiny. And the space between the alveolus and the capillaries is less than half of a micrometer. And this allows oxygen and carbon dioxide to be able to easily exchange over that short distance. The second type is type 2 pneumocytes, or AT2, and this is more numerous. They're more common than type 1. About 90% of the cells are this type 2, and they occupy about 5% of the surface area in total. They're about 10 micrometers across, and they have many organelles within the cytoplasm, including mitochondria. The large amount of phospholipids are synthesized in the cytoplasm and are stored in these lamellary bodies, and these vesicles and vesicles with multiple layers of phospholipids and proteins are excreted by exocytosis. Muscles provide the ability for movement, and that movement is created by one muscle being pulled to shorten in length, to contract, and that thereby exerts a force that causes the movement to be able to return to its before state, its resting state. Another force must be exerted on that muscle, and that usually comes from another muscle. And so muscle cells have some specific adaptations to be able to allow them to accomplish these different movements. Skeletal muscle is muscle that's used to move the body, and it's attached to bones. It can also sometimes be called striated muscle cells, because when they're viewed under the microscope, they have stripes. They are long, unbranched, cylindrical structures that are arranged in parallel groups. They have multiple nuclei, and they are much larger than other cells. They contradict the cell theory of what we would expect to see cells in terms of their size. How they get so much larger, increase in size, is caused by fusing of the embryonic muscle cells. Actually fuse together, and they're larger and have more nuclei as a result. Myofibrils are many parallel cylindrical structures with alternating light and dark, and they have a disc-shaped structure within them, and that light band is the Z, referred to as the Z line. A second type of muscle cells are cardiac muscle cells, and these are muscle tissue that you would find in the heart. They form the wall of the heart, and it also has a striated appearance. However, they're much shorter than skeletal muscle, and they only have one nucleus, so they're smaller and don't have as many nuclei. And within their points of contact, they have an interconnected disc that allows branching of the cardiac muscle cells. So the skeletal or striated muscle are parallel bundles, but the cardiac muscle can have branching, and this connection allows the connections then allow connections between the plasma membrane and cytoplasm of different cells, and this is important because electrical signals need to be sent through those cells, and that is what controls the heartbeat. And so these would be two different types of muscle cells that have adaptations to be able to carry out their function. Our last cells to examine are the gametes of humans and other organisms, and that is the sperm and the egg cell. And these are cells that are haploid. They have half the number of chromosomes. So in humans, this is going to be 23 chromosomes each. And looking at the egg specifically found in females, its function is to receive one sperm during the process of fertilization. And the zona pellucida is a layer of glycoproteins containing ZP3 that the sperm bind to and can penetrate. And it can be chemically altered at a later point in time to be able to prevent additional sperm from being able to fertilize the egg. The binding proteins of the plasma membrane enable fusing with sperm and actually allow it to be able to enter the egg. It also has corticola granules, and these are vesicles of enzymes that are released into the zona pellucida via exocytosis. that make the egg impenetrable to sperm after it's entered the egg. It blocks additional sperm from being able to enter. The yolk structure of the egg itself stores lipids and other food sources in the cytoplasm so that the developing organism, the zygotes, the fetus eventually has an initial food source. There's lots of mitochondria within the egg to produce ATP, and actually all of the mitochondria within an organism come from this initial egg cell, not from the sperm. So all of your mitochondria are from your mother. And additionally, or lastly, it also has centrioles, which are used for cell division. In the process of mitosis, it helps to control that process. And so these would all be different structures that the egg cell has to be able to allow it to receive sperm and then fuse with the sperm, and specifically the DNA within the sperm, to create an offspring. The purpose of the sperm then, its objective is to be able to transfer its DNA to the nucleus of the egg, and it's a competitive process. Males produce billions of sperm throughout their lifetime, and it's numerous sperm that are released in order to try to fertilize the egg, but only one actually is able to do so. The sperm cell itself is much smaller than an egg. However, it does have a very long flagella, the tail, and the tail is arranged in 9+2 microtubules, and the purpose of those is to be able to generate force for swimming. The mid-piece portion of the sperm has much mitochondria in order to be able to provide ATP for the movement of the tail, and then the head shape of the sperm, the head portion, is streamlined and narrow, and it's tightly packed to be able to minimize resistance. The chromosomes within it are tightly packed to minimize resistance. It has receptors to be able to bind with the ZP3 in the plasma membrane of the egg. It also has an acrosome, which is a sac of enzymes that help to digest the proteins and polysaccharides that are in the protective zona pellucida. And lastly, it has binding proteins to be able to bind to the protein in the plasma membrane of the egg and allow fusion and entry of the sperm into the nucleus. And so just like the egg has a number of specific adaptations to be able to facilitate this process, the sperm does as well. And so this concludes looking at the different types of specialized cells with specific adaptations to be able to carry out their function.